So hello everyone, welcome back to my channel Meiosis. Today I am going to discuss about the Partussis Toxin Pathway, which is basically the extension of the GPCR pathway, right? Partussis toxin is actually abbreviated as PTX, so we can call it PTX. It is basically a exotoxin and it is actually secreted by the gram-negative bacteria which is termed as Bordetella pertussis, and it is a gram-negative bacteria. Okay. So when the Bordetella pertussis infects us, okay, it will release the PTX toxin and it will target our respiratory tract. Basically the respiratory tract epithelial cells, right? It will generate a disease condition which is known as whooping cough. So how it will do that whooping cough? So basically it will destroy the ciliated cells of the respiratory tract epithelium. Okay. It will inactivate the NK cells, also known as the natural killer cells, and phagocytic cells. And number 3, cellular dysfunction of the respiratory tract will occur. Okay, so this is the symptoms of the whooping cough. So let's see the structure of the PTX, right? Because before going to the working mechanism, we should know the structure of the PTX toxin. Okay, so basically it is a AB toxin. AB toxin. More prominently, it is a AB5 toxin. The A subunit is called S1 and B subunit is composed of the 5 subunits that are from the S2 to S5. Right here, the S4 is in the S4 is basically 2 in number, right? So basically it is like that. Here is the S1, the S4 is 2 in number, and others are the S2, S3 and S5. This is the A subunit, this is the B subunit. Okay, so basically the A subunit, which is the S1, is capable of ADP ribosylating. ADP it is actually capable of the ADP ribosylation because it has the ADP ribosyl transferase activity. Okay, but the B subunit is actually helps in the binding to the receptor, and the PTX will bind to the asparagine linked oligosaccharide and lactosyl ceramide sequence, right? And this PTX toxin producing gene is present in the chromosome of the Bordetella pertussis bacteria, right? So this is the structure of the PTX. Okay. Now we have to know about the working mechanism of the PTX. Okay. So let's see the working mechanism of the PTX. The working mechanism, right? So basically, Partussis toxin, by the help of the B subunit, it will bind to the receptor. Okay. And in a normal GI pathway, in the normal GI pathway, it will do the ADP ribosylation. In the cysteine residue of the GI alpha subunit, right? So basically in the normal condition, the GI will inhibit the adenylyl cyclase enzyme. It will inhibit the adenylyl cyclase enzyme, right? And also the PK production and also the CAM production, obviously, right? In the normal condition. But when the PTX toxin is actually released from the Bordetella pertussis and it will bind to the receptor, so what will happen? The PTX toxin is actually do the ADP ribosylation in the cysteine residue of the GI alpha subunit, and the GI alpha will be locked in the inactive state. If the GI alpha is locked in the inactive state, so it cannot inhibit the adenylyl cyclase and the PK and CAMP will be produced. So the GI alpha inactivated GI alpha cannot inhibit the adenylyl cyclase and PK CAMP production. So the more adenylyl cyclase enzyme will be activated, more more CAMP will be produced and more PK will be generated, right? And CAMPs are also helps in the chlorine influx in the respiratory tract epithelium cells. So because it is the ligands of the ligand gated ion channel, so the chlorine influx will lead to the mucus secretion. So the body will produce a jerk, a mechanical jerk to remove the mucus from the body, and the whooping cough will be produced. Whooping cough will be produced. So this is the disease condition which is known as the whooping cough, and this is the working mechanism of the Partussis toxin pathway basically. And I hope you all understood the mechanism of the Partussis toxin pathway. So for today I'm going, and I will meet with you in the next video. So bye, take care.